Hello, my name is Pedro Oliveira. I'm a master's student from the Faculty of Science of the University of Porto, and today I will present my thesis project, Arginine vasotocin regulation of social behavior and dominance in the common waxbill. So, arginine vasotocin, also known as AVT, is a neuropeptide that has been found to influence the behavior of multiple species of animals in various ways, with an emphasis on sexual and aggressive behavior. These effects have been recently studied on a large group of bird species. Interestingly, however, these results were not consensual between species, with some birds acting more aggressively when infused with the compound, and others reducing their aggressiveness. These studies also tended to show differences between sexes, with males often demonstrating a more pronounced effect to the presence of AVT than their female counterparts. For this study, I aim to document the effects of AVT on the social behavior of the common waxbill, a highly gregarious passerine indigenous to southern Sahara and with a very strong presence in the Iberian Peninsula. So, in this experiment, the birds were first food deprived for 1 hour and 40 minutes, after which one pair of individuals, one male and one female, received an injection of one of 4 treatments, low and high dosages of AVT and low and high dosages of manning compound, a specific antagonist to one of AVT's 3 receptors, V1A, while the other pair received a control treatment. After the injections, the animals were subjected to another 20-minute fast, followed by a food competition test using a feeder in the middle of the cage, during which they were recorded for 15 minutes. For this, 24 individuals were gathered, 12 of each sex, and housed in 6 cages, 4 birds each. Each week, the compound and the pairs that received them would be swapped, until every individual received each treatment once. Furthermore, in one of the test weeks, all the individuals of the cage received control. After the tests, the footage was analyzed to determine the variance of 5 different variables: movement, feeding time, latency to feeder, aggressiveness, and allopreening. A principal component analysis was performed to link the variables feeding time and latency to feeder, turning them into a feeding PC. Lastly, the data was treated with general mixed models to determine the effects of the treatments. The results showed a significant reduction of all the variables when the individuals were treated with the high-dosage AVT injections. Individuals also showed less movement when treated with the lower dosage AVT treatment. The analysis also showed that males tended to be naturally more aggressive than females, which led me to perform a separate analysis to determine if the 2 sexes reacted differently to the treatments. And there was indeed a significant difference in aggressive behavior between males and females after receiving the low MC injections, with the males increasing their aggressiveness but not the females. So, in this experiment, I determined that AVT heavily influences all the studied aspects of social behavior in this passerine, being this influence likely linked to other receptors than V1A, due to the lack of changes experienced in the antagonist treatments. Furthermore, through the use of manning compound antagonist, I showed that the blockage of V1A pathways significantly increases the frequency of aggressive behaviors in male individuals. These results are in line with some of the previous studied species. However, as mentioned in the introduction, those studies were not consensual, meaning that others go in the complete opposite direction, with AVT increasing the aggressiveness of the individuals. Furthermore, this study was also able to demonstrate the involvement of V1A pathways in the aggressive behavior of the common waxbill. These results seem to corroborate the idea that AVT is, at least in some way, necessary for keeping the levels of aggressiveness to a socially acceptable level in this bird. Lastly, I would like to thank my supervisors, Trigo and Soares, as well as all the people involved in the behavioral ecology group that welcomed me so well. Thank you.